Coleorrhiza. Coleorrhiza is a undifferentiated sheath which is protective in nature and protects the root or the root cap. Similarly, you will find in the monocot embryo coleoptile, which protects the plumule or the shoot apex. So these are some major differences between monocots and the dicot embryo. These topics are already covered even in your 11th class syllabus. So students, I think you would have understood now that post-fertilization events involve a lot of changes in the plant. The major change is the formation of the endosperm. Without endosperm formation initiation, zygote cannot divide. So zygote developing into embryo starts later, endosperm formation starts earlier. An endosperm can be whether either the free nuclear type or the cellular type or the halobial type. If it is a free nuclear type, there will be no cytokinesis, only karyokinesis, and a large number of nuclei are formed. Coconut is an exception where you will find that free nuclear endosperm slowly changes into cellular endosperm. So liquid form changes into the solid form. The white portion which you consume in the coconut is the solid endosperm. The liquid which you drink from there is the free nuclear endosperm. This gives more energy compared to this one. Cellular endosperm is the one in which every nuclear division is followed either by a transverse or by a longitudinal plane of cytokinesis, and finally a cellular type of endosperm is formed. Halobial is a type in which you will find that the first division is an asymmetrical one, a larger cell towards the micropyle, a smaller cell towards the chalazal end, and this cell will basically give rise to a free nuclear type of endosperm. So first division is cytokinesis following the karyokinesis. Other divisions are without cytokinesis. This may develop into an hostorium, most of the monocots. And the formation of embryo starts from zygote. Zygote first of all divides into two cells. One cell is the basal cell, which is the suspensor cell, which will give rise to a long suspensor later on, and the bottom cell, here it is called as the apical cell, is the embryo cell, chalazal end and the micropylar end. Please remember that. So the embryonal cell will divide by a vertical plane and the suspensor cell will divide by a transverse plane. This cell will develop into hostorium. This will divide a number of times to form a series of cells. The whole thing together is called a suspensor. The embryonal cell divides first by a vertical plane in one plane and then the other plane to finally form 2 cell to 4 cell situation. Now it is a transverse plane of division. Now 8 cells are there. Then a periclinal stage of division, so 8 cells and 8 cells, total 2 rows will be there, so 16 cell stage. The outer cells out of these 16 will give rise to epidermis, and inner will give rise to stele and the pith as well as the cortex. Up to this point it is called as proembryo, up to 16 cell stage. Then onwards it is called as an embryo. Embryo first is called a globular embryo, then a heart-shaped embryo, and then a mature embryo. The mature embryo of monocot and dicot is different. Monocots will carry only one cotyledon, and in the case of grasses, that cotyledon is highly reduced and it is called as scutellum. In case of dicot, you will find there are 2 cotyledons. They are well developed and because they are storing the food. This one is minimum amount of food storage. The second cotyledon does not develop in the monocots embryos. It is referred to as epiblast. There is no sheathing, protective sheath around the plumule and around the radicle in the case of dicot plants, but you will find there is a coleorrhiza and a coleoptile which covers the shoot apex, future shoot apex and the future root apex. I hope students this topic is clear to you.